All right, great. Thanks very much to Janan and Sean for inviting me. This has been a tremendous amount of fun interacting with so many different disciplines here. So we all know that evolution is a natural selection of favorable traits in a given environment. And today, in my talk, I want to explore how it is that aquatic versus terrestrial environments might push or favor or disfavor certain kinds of approaches to action control in animals, focused on vertebrates, though there'll be a shout out to invertebrates at the end. Here's three different prey-capture sequences I've collected over decades on different animals. On the left, you have an electric fish hunting a water flea in complete darkness. In the middle, you have a larval zebrafish, 4 millimeters long, hunting a single-celled animal. And on the right, you have a dragonfly nymph hunting a larval zebrafish. Now, I've frozen all of these videos at the frame that we estimate detection occurred, where the predator starts to react to the presence of the prey. And the thing I want to point out is something super basic that everybody who works in aquatic predator-prey relationships knows that dynamic interactions happen in near field conditions in the water, typically not uncommonly at less than a body length. I'll play these just so you get a sense of reactive control at work. These are all high speed, all high speed interactions occurring with very low latency. Now, why don't we just stick with that? This is a perfectly good way to operate action control, very high speed advantages to that. But the problem with high speed is that it's highly stereotyped. And when you have highly stereotyped low latency responses, you can have your nervous system hacked by a smarter predator. And that's what I want to show you next. Some video by Ken Catania on this is a tentacled snake. These are snakes. They obviously evolved on land for a long period of time, and this animal is secondarily aquatic and now is largely purely aquatic animal. And what it makes a sport of doing is triggering the escape response of this animal through rippling from a tail to head, very, very low amplitude muscle motion that you can barely see in the video. In fact, I don't think you'll see it. Is it triggers a pressure wave of water, which causes this animal to initiate an escape response. And it's so predictable, you can see exactly what happens. And boom. So pretty easy eating for a terrestrial vertebrate coming back to the water. Now let's contrast those short, high-speed reactive responses to a typical stalking sequence you could see on the savanna. Here I've clipped off a large amount of time where it's stalking along this bank, this leopard, and I'll just show you the end of it, where it's exploiting a pencil-thin shadow of occlusion to its advantage. Something that is readily present on land, but not so useful in water. You can see it, the antelope's starting to get paranoid, something's going on, but it doesn't see it. And now all that the cat has to wait for is the next munch, a little bit more damage. And there we go. So I won't show you the rest of that, but you can predict how this turns out. So this is a long sequence, right? Many body lengths over a long period of time. So let's just cartoonize this arrangement. But first, before I do that, I wanted to mention this work we did looking at what is the fundamental basis of this disparity between this near field control and far field control. And I would argue it comes down to something that's super basic, which is that air is transparent to light and water is not transparent to light. So I got very curious what happened to visual systems at the water to land transition 380 million years ago. And myself and a paleontologist, Lars Schmitz, went back and we did a lot of measurements from fossils and uncovered that there was a tripling of eye size at the water to land transition. And why didn't it happen before the water to land transition? Well, we did a bunch of simulations of image formation in water versus land of a 30 centimeter black disk. Okay, and these were very careful simulations with full, very careful optical models of water. And through a coastal water model, we get a detection distance where you can just make out the disk of 7 meters at the ancestral eye size. This is the head of Euthanopteron that lived 385 million years ago. And then this is Acanthostega, 350 million years ago. Its eyes have tripled in size. And now through water, the increase in visual range is basically negligible. But in air, we get a significant jump. And on net, if you combine the medium change plus the change in the eye size, you get a million-fold increase in the total visual space that the animal can interrogate. And so we have osteological correlates of eyes that are very that we can interrogate with the fossil record. We don't know what happened to brains, right? So one thing we can say is that I skipped forward a little bit. Let me first, before getting to brain size differences, let me show you kind of a schematic of how we're thinking about these two different spaces. The aquatic space with a blurry visual scene, and on rare occasion you might see an animal in front of you. You're a prey with a small sensory bubble going through space. If you're fast and you kick off, say, your Modner cell and you get a 10 millisecond gain in escape response, maybe you can escape to live another day. And on land, you get an entirely different situation. From this perch, we have very long sight lines. Of course, if you're in this cluster of trees, you're going to have a mix of short and long sight lines. And so in this space, but not in the aquatic space, it seems evident that if you evolve the right set of neural structures, you could potentially evaluate some possible futures, such as this one, which is not so hot, this one also not so hot because you're going to have to jump through water, swim through water, and then this one, which is clearly the best, and then with some evaluation circuitry and some inhibition of motor outflow to prevent yourself from doing the first thing you think of, maybe you can actually execute that best strategy. So that's a lot more processing, and so here's where I get to the note about brain size that I wanted to mention. So a little appreciated fact is that warm-blooded land animals, birds and mammals, have a 10 times larger brain to body mass ratio than ancestral aquatic animals. And so here's a goldfish brain with a 0.5 centimeter scale bar. Here's a whale shark, 2,200 times the size of a cat. And here's a cat brain. So that tells you at a glance what we're talking about. But you can measure this across many different species, and there's no mistaking a big jump in brain size occurs with the warm-blooded animals. So we wanted to start getting some insight into this using modeling. And so what we did was we developed some software, Grid World, where we have a predator, we have a prey, and occlusions, which are black here. And then for planning, we give the prey agent the ability to plan. And the way we do this, you can just think of this as building trees of possibilities using something called Monte Carlo tree search, which is just a way to do that tree growth without becoming a victim of the unfavorable combinatorics. And so you can do this and build out your trees. And you have to deal with the fact that in some cases, the prey will lose sight of the predator. And so what do you do then? And so there's a particular framework, either using Bayesian filters or something called a particle filter. We use a particle filter to model the belief state of the prey agent when it can't see the predator. All right, so with all that infrastructure, we can also just have a knob to dial the amount of planning from a little bit of planning where we only have 10 forward states simulated to here we have 100 states forward simulated. The peak that the algorithm uses is 5,000 states forward simulated, and so we're going to compare behavior at 5,000 states forward simulated to our model of reactive control, which is basically taking the most successful strategies over many, many episodes and canning those and unfurling them as a function of applicability to the current situation. When we do that, two things pop out. One is the survival rate is quite low. It peaks at about 10% survival rate at maximal visual range. So we're doing the aquatic thing of low visual range up to 5 body lengths. One body length is one here. So up to 5 body lengths. And we're doing our planning, and there's no obstacles in the environment. It's open space, so it's kind of like a aquatic-like environment. And we get no significant difference between the reactive algorithm and the planning algorithm. Now we go into a different mode of modeling here. Now we're going to actually add clutter. So we have a metric for clutter and a way to randomly generate hundreds and hundreds of worlds for statistical robustness. And so I'm going to show you now how planning efficacy varies as a function of terrestrial-like clutter in this very idealized environment. So here we have 100 states forward simulated, 1,000, 5,000. And look at the signal in the mid-clutter regime between 0.3 and 0.7. We get, not only do we get a big boost in survival rate, remember 10 was the highest we had before, but now we have unlimited visual range of a very small world. That's very reasonable in this very small world. And we get up to 40% survival rate, and we see a big effect of planning at different levels of forward state simulated. And so let me show you some of the trajectories. So at low entropy, at low clutter here, survival rate is quite limited. The trajectories we see were almost exactly like what we saw in our aquatic-like scenario, which is they went along the wall. And this is actually a well-known phenomenon amongst animal behavioral experts. This is kind of a trace of anxiety to the point where people who test drugs for anxiety will look at how much an animal will hug a wall during behavior in an open field. And so in this open field, we actually were surprised to see the emergence of this behavior called thigmotaxis. And the success paths, there weren't many, but the success paths always hugged the wall. Now, in the high entropy condition, planning also has negligible impact. I don't show this data, but in both these low and high clutter regimes, the advantage over the reactive controller is not significant. And you can sort of see why here, that essentially the survival rate depends, or whether you survive on a particular trial depends on where this predator spawns. And that's a random thing. We just randomly seed that. And so if you spawn very close to where the prey is, which is always in that center square at the bottom, then the prey isn't going to survive. And if you spawn far away from that point, the prey is going to survive. The characteristic survival strategies in this success paths in this environment are also pretty stereotyped. But so let's look at the mid-clutter regime. And what we see here is the emergence of really, I think, quite fascinating strategies. Here's one where the prey goes into this little sheltered area and escapes out this opening on the side after the predator has come close enough that it's secure in the knowledge that it's going to be able to shoot out the side and get to the goal. So we see lots of these kinds of strategic patterns or seemingly strategic patterns of behavior in the mid-clutter regime, and not in the low or high entropy regimes. The success paths are now quite a bit more diverse. They're not stereotyped. So obviously these simulations they're 15 by 15 grid world, there's turn taking, there's all kinds of idealizations that obviously might fail to be supported in terms of mapping to reality, biological reality. So we need experimental support. But the grid world was too small for live animal experiments. So we made a bigger arena, hexagonal, and we developed a fully automated paradigm for measuring strategic evasion in a predator-prey-like task and comparing it to simulations as well. And here we're going to use a rodent. I don't know how to work with mammals, so being a fish person, so I paired up with Dan Dombeck, who is a master electrophysiologist with rodents and a master imaging person as well. And he works on the hippocampus, which is a key structure for these behaviors. I think I'll show you a bit of that later. And so we have a project now ongoing for a few years now, and a couple of postdocs I want to mention who are integral to the data I'll show you are Chris Angeloni for some of the behavioral work and neural pixel recordings of brain activity, and then Herman Espinoza for simulations and mechatronics. And we've had three tremendous PhD students who are integral to all of the data: Alex Lai, Orjan Mugan, and Gabby Wink, who's especially done a lot of great work on the robotics side. So here's our system. It's a water reward port at the start and at the end. These are water restricted animals, are highly motivated to get to their water rewards. And we have a robot here. So this robot is an autonomous robot. It isn't controlled by us. All it does is look out and looks for the mouse. If it sees it, it chases it. If it doesn't see it, it just looks for the next cell that it can't see behind and randomly goes there. So this robot has kind of an interesting developmental history. I want to show you a little bit of that in a second. But first, I'll just tell you a little bit about the arena, which is got magnetic obstacles for rapid reconfigurability, so we can test a whole bunch of different types of environments. And so here's the life history of the robot I'm alluding to. So I thought when we first started designing this robot that the robot itself would be sufficiently aversive to not require any specific aversive stimulus, that just this big lumbering robot would be something the mouse would be scared of. And it turns out that I was quite wrong. And in fact, what we'd invented was the first rodent Segway. So take a look at this. So it jumps on the robot after a short period of time. And this is the first rodent surfing video ever. Could be a new Olympic sport, I don't know. But in any case, we were pretty surprised to find this. And so we had to add an aversive module to the robot. And so we did. It's a CO2 blaster. This is not an air puff. This is a blast of air. And you'll see the effect. So when this arrow turns red is when this animal will get blasted, okay? And so you'll see its response in a sec. And so I'm a little bit surprised it turned back to look at the robot, but I assume it just thought it wasn't fast enough to catch up and just wanted to see what the hell was going on before escaping. But anyway, after that first exposure that a mouse has to this puff, they generally avoid it like the plague. So it is sufficiently aversive to motivate evasion. And so here's our task. It's called bot evade for obvious reasons. You start at the start port, you go through the space evading the robot and its menacing puff, and get to the end port, which also has a water reward. And then this brown chute here is a return chute where it can safely proceed back to the start for another trial. So we get thousands and thousands of trials of data, all automated. And I want to show you a behavior that all the mice quickly learn, which I find fascinating. Some of you are probably aware of something that birds do called the broken wing display. And in that display, basically a ground nesting bird will see, if they see something like a fox approaching the nest, they'll come out of the nest and pretend that they have a broken wing and walk away, and until the fox is far enough away from the nest that they can then fly off and basically laugh at the fox. So we see something very rapidly emerge in all the mice, which I think is reminiscent of that. And you'll see it here in a sec. Basically, they go up to the robot and then lure it back. And then once they know it's over in this corner, they just run along the north side. Okay. So every mouse learned this. They learned it very rapidly. It's like they seem to maybe know that they can get away with this. I don't know. But another behavior that we're fascinated by is use of the leveraging the occlusions. So you'll see now in this trajectory a few points at which the mouse comes up to an occlusion, looks at the robot, pauses, and then reroutes. Okay, so these are very exciting events to us. We now have probes in these animals recording neural activity to see if we can see neural correlates of planning at those points, and you'll see some of that data in a sec. So here's some of the data. Here's without robot trajectories colored by speed. So red is high speed. The speed goes down and the trajectories get a little more diverse with the addition of the robot. If we then take out the robot, a lot of the diversity just quickly collapses. They quickly are in the know that there's no risk, and they just book through the space for their water reward. With no obstacles, there's the trajectories are pretty well with no robot present. They're quite widespread. But as soon as you add a threat, we get highly thigmotactic, uninteresting trajectories. And so I really want to highlight. Think about the largest biomass on the surface of the Earth is plants, really only present on landforms, and this is the occlusive environment provided by plants. I think may be integral to some of the neural control complexity that we see in the brains of vertebrate land animals. So we clustered the mouse trajectories, and we looked at things like the number of clusters and the mean distance to the nearest cluster. And we see that the number of clusters goes up with the addition of the robot. Post-robot, it collapses, and the distance between any given trajectory and the nearest cluster goes up significantly as well with the addition of the robot, and it again collapses. So these are measures we're using as a proxy for the complexity of neural control of action. And I want to show you this trajectory because I think it's really illuminating. This is the first exposure of this rodent to a robot that didn't move, just sat there quietly. And the puff was deactivated. It just sat there. And take a look at the reaction. The velocity is here, but really the behavior of the mouse is quite interesting. And I think it highlights not only may mammals have a superpower for the acquisition of internal models, it's the dropping of an internal model when it no longer applies. It's pretty fascinating here. So it keeps going back and forth. Wait, I haven't baited it yet. It's still not chasing me. I'm going to come back, try again. And this is its last time. It gets really bold, and then at the end, it chickens out. And now it does something completely different we haven't seen it do before, which is proceed very carefully with a lot of vigilance, a lot of head scans in the direction of where the robot might be approaching from, and just carefully goes along the edge of the wall until it gets to its water reward. All right, so we can synthesize now the earlier modeling infrastructure with live animal behavior. And that peaking trial I showed you a few slides back, we now just implement the particle filter running off the kinematics that we recorded from the live mouse to try to get an idea of what kind of theory of mind might this mouse have with respect to the dynamics of this threat. So I don't have a lot of time to go into the details here, but I do want to highlight that last evasive maneuver at the end. You'll notice it's just outside the radius of diffusion of the robot, at least of the belief state of the mouse of where the robot is. And we're also showing the view space of the mouse over time. All right, so we're analyzing a lot of data like this right now for trying to get insight into how much does this optimal approach to belief estimation map onto the animal's actual behavior. Now, whether an action is planning-based or cached action recipe, habit/reactive, it's clearly not going to be resolved just by looking at behavior. Behavior is going to the neural implementation will be underdetermined by the behavior. But there are putative neural correlates of planning that we can begin to examine by recording neural activity during the task. And so that's what I'm going to show you a little bit of now. But first, I need to tell you a little bit about one structure which is a good candidate for looking at neural activity, and that is the hippocampus. And the hippocampus contains something called the cognitive map, which is essentially if you record one cell in the hippocampus, it'll light up when the animal's in a certain position in the arena. And this is, I should say, not dependent on body orientation or head orientation. This is with respect to world coordinates. And this was discovered some years ago. Nobel Prize was won a few years ago for this. And basically, you can figure out with more recordings, that the entire space is tiled by the receptive fields of cells, of place cells that report whether or not an animal is in a given location. So the brain has a way of knowing where it is and then processing further details based on that estimated location. So now what you can do after a bunch of neural recordings is build up a decoder, which will basically let you in real time estimate where the animal thinks it is in space and compare that to where it actually is in space. And so I'm going to show you some of that data, because one of the things that other investigators have started to see is that most of the time the place cell is activated for where the animal actually is. But then there are these crucial moments at key decision points where the animal will stay stationary and you'll see a drifting of the representation of where it thinks it is in space to different locations, in particular places it might want to go or has been in the past. And so these are called preplays or replays, also called sharp wave ripples and theta sequences. There's a hodgepodge of these phenomena that are putative physiological correlates of planning that we want to start looking at. And so I'm going to show you some, first of all, evidence that we can find place cells in this task. And so we have the mouse running around our hexagon with the robot chasing it, and we can see place fields according to where the mouse is in space. But we also see something very cool, which is place fields representing where the robot is in space that are stable over time. So in this particular case, whenever the robot's in this part of the space, that cell lights up. Okay, so there's a representation available for the dynamic threat that the mouse is facing. And so now I want to show you one of these decoder videos. So this is a slightly different task from bot evade. Instead of a start and end water reward, these are water ports. We did this for a technical reason having to do with coverage of the space. But so these are water ports, and the animal's trying to get to these water ports. It doesn't know which one is turned on, and it's being chased by the robot. So here's the robot and its actual position. Here's the mouse and its actual position. And the decoder is that pixelated thing in the background that you'll see move in a second. And so you're going to see that most of the time, the place cell for the mouse is where the mouse is, but then it drifts. So right here, it doesn't yet see the robot approaching from the lower left or lower right. And now it does see the robot approaching. And you can see patterns of activity to these two foraging locations, this one and this one. And eventually a decision occurs to go to the one further away from the robot. So this could be, in this corner, in other words, some neural analog of planning going on. So I'm going to shift to something a little bit different, which is starting to look at humans. So if planning is an early tetrapod innovation, say 250 million years old, it could be, that's the emergence of the first warm-blooded animals that start to get these big brains. There's a question, what do we share with other mammals then? Is there a commonly inherited core circuitry for planning that we might have inherited from our last common ancestor to mice? That was 90 million years ago. And so to look at this, we've devised a virtual reality task, which is a free body task where the participants are wearing a headset and they're in a replica of the mouse environment with the same obstacles in the same positions, just scaled up a whole bunch. And so I'm going to show you one of the trajectories that the people did in this space. I got to say, this first one. These people have more faith in our technology than we do. With those columns so nearby. Like, we're doing it in a much larger space with no columns right now. So, but anyway, so now we can do direct head-to-head comparisons of the trajectories of mice and humans. This is just a small amount of data. We have a lot more now we're analyzing. To look at, are the pause locations the same? Are the peak locations the same? What are the evasive strategies that might be common or different between humans and mice doing this task? All right, so I've got just a couple more slides to finish up. So if planning visually guided behavior depends on having long sight lines mixed with short, then it could occur in any animal with good visual range in the mid-clutter regime. Now there's this spider, Portia. It's a genus, few different species. Labiata is the one we look at, Portia labiata. It's a jumping spider that has this unbelievable visual acuity. It's similar to a pigeon's visual acuity, and that's an order of magnitude higher than any other terrestrial invertebrate. So they engage in hunting sequences that last 30 minutes to 2 hours, and these hunting sequences are just unbelievable, and they kill venomous spiders that can easily kill them. So this is a very dangerous kind of hunting sequence where the hunter could easily be killed by its prey. So could studying planning in this animal show us some principles of convergent evolution of planning between animals whose, in this case, last common ancestor was 650 million years ago? So clearly not a planning animal. So here's a picture I took in the Malaysian jungle last summer. Here's an acuity plot, and here are my collaborators, Daqin Li in China, Chen Li in Johns Hopkins here, and Jimena Nelson in New Zealand. So we have a grant to do this work. We just started work not long ago, and we've just done one field trip, and now we're building laboratory apparatuses to start to automate high throughput behaviors. So I have a video sequence. I was unbelievably lucky enough to have my iPhone in the right place at the right time to actually get a full hunting sequence. It lasted 43 minutes. And it took about over about a meter for the animal to get to its prey. But it's too long to show you here. I can show you a highlight reel another time if you're interested. So I'm going to have to skip some new reinforcement learning work, but just give you the headline of it, which was we did some comparison between the mouse behavior and a reinforcement learning agent controlled by state-of-the-art algorithm, TDMPC2, and you can see that it does very, very different trajectories. And the problem, as people in reinforcement learning know, is that reinforcement learning agents don't care about dying. In fact, it's a good signal for training the neural network. And so we had to try to figure out, how do we get the fear of God into these reinforcement learning agents. And we did so through two different mechanisms I don't have much time to go into, but essentially one of them, you could think of it as inducing post-traumatic stress disorder. We simply played negative experiences from the training set over and over and over to the algorithm to get a heightened sense of the negative possibilities of their action. And we did another trick having to do with the variance of the Q table. But I don't have time to go into it, but we got significantly better coverage of the space over what we had before. So we're excited about this. Is a very different approach. Remember, the first algorithm I showed you, we actually had to give the internal model to that algorithm. it wasn't learned because we're focused exclusively on planning. Here, everything is learned from the ground up. So it's a very different modeling approach and one we're excited to dig further into. All right, so that's what I just said. So visual sensory volumes grew by about 1 millionfold after landfall in the Devonian, starting 385 million years ago. Distal sensing generates more time for neural calculations. Model-based planning and simulation appears to only be advantaged with the partial visibility of mid-clutter terrestrial environments, and this could be, therefore, a geological constraint on cognition. So if Earth had been a water world, it seems likely nothing interested in being an audience member of a symposium on natural philosophy would emerge, and apologies to the octopus out there. But I do have comments on the octopus that maybe we can get to in the discussion section. Early results from a new paradigm to test this idea experimentally with future comparisons to humans and spiders. And then porting the task to RL is highlighting the role of irreversibility or mortal threats in learning, which are not present in the standard RL implementations. Okay, with that, thank you for your attention.
Well, thank you very much. Thank you to the organizers of this symposium. It's great to have this, perspective with different aspects of cognition, philosophy, mathematics, computations. Thank you, Malcolm, also for this provocative perspective in how environment shapes and helps the evolution of cognition. This talk was particularly inspiring for me, given the seamless connection between ecology, modeling, behavior, and physiology, ultimately converging on how strategic behaviors emerge from environmental constraints. I want to use my time today to reflect on a few key points that you've raised and to expand this perspective with some interesting phenomena that we have observed in our lab. I will be presenting a series of questions that I have all around these three general ideas that relate to Malcolm's presentation. First, you began your talk by a compelling geological argument for the emergence of intelligence, that long-range satellites in terrestrial environments expanded visual access, allowing time for plant-based cognition. You observed that survival peaks specifically in mid-clutter environments where animals are prompted to explore, pick, or bait, like we saw with the mice, and benefit from planning strategies. But in fully open or fully cluttered spaces, the environment is either too transparent or too opaque for planning to be beneficial. So I'm wondering whether it's not simply the quantity of sensory information that we have in the environment that drives the evolution of cognition, but rather the structure and the variability of that information and how it's distributed in the environment. Is it the presence of just enough occlusion and variability that enables animals to infer and track predictable sequences where planning becomes not only possible but advantageous? Do you envision a way to formalize the relationship between information streams and effective distribution of clutter? Do you think there is a threshold at which sensory uncertainty becomes just right for prospective cognition to emerge? All these questions also give rise to a more general idea. Could prospective cognition be just, generally speaking, not a visually dependent aspect, but generally a sensory, it could be generalized to other sensory systems? For example, I'm thinking about animals like echolocating bats that are navigating clutter environments at night, for whom seeing is not really seeing. And they basically have a completely different structure for predation. Here, the general question is outlined in number 1. Is planning a modality-independent response to environmental uncertainty rather than a vision-specific adaptation? Could these cognitive strategies seen in your visually guided task parallel to other sensory systems? For example, I'm curious as to if anybody has tested how blind mice are performing the bot evade task. Would they develop a modality-specific planning strategy based on hearing and somatosensation, for example? Are their trajectories also revealing a different relationship between environmental structure and cognition? And I think some of these questions can help clarify whether prospective cognition depends primarily on what is sensed in the environment, or how and where the information is actually structured in space. Second, you emphasize how environmental constraints drive the evolution of cognition, and made a strong case for planning across the spatial realm. One of the first things that came to my mind when I saw your bot evade task was this fascinating book I came across a few years ago called Mice All Over by Peter Crawford. He was a scientist appointed by the British Ministry of Agriculture to study mice trajectories in grain storage facilities, aiming to better understand their movements and improve pest control strategies. He began by observing individual mice in simulated spaces like the ones you just showed in your presentation, except, of course, this was before the era of automatic tracking algorithms. So he literally sat on a platform for hours with a pencil and paper, hand drawing the movements of mice day after day. He then gradually added more mice until the environment reached a full group. And what he found was really interesting. When housed in larger groups, mice would self-organize into smaller subgroups of varying sizes, each occupying distinct, non-overlapping nesting territories. And when a mouse from a group crossed into the territory of another mouse, fights would break out. So linking this with some of the work that's happening in the lab I'm part of, the Cuchibotla lab here at Johns Hopkins University, we use what we called a continuous learning playground. And this is an environment for mice where they live in a home cage, but they need to enter a behavioral box to complete a task to get a water reward. These are also water-restricted mice. This is a great setup, not only because it allows us to obtain extremely cute pictures and photographs of mice, but we can also assess and understand very interesting underlying social behaviors. For example, we have seen in the behavioral box that there are short-range interactions between mice that are due to competing for environmental resources, like this. In this case, these two mice are competing to drink water from the water spout. So we have this short range pushing of the individuals. We have another example in the home cage box. This is where the animals live, where an animal actually took too long in the behavioral box, and when it came out, of course, all the other animals in the group were waiting. So the dominant mouse of the cage started a fight with the one that was inside, as a way of saying that that animal was taking too long and the others were waiting for water. So between what we observed in this playground and Crawford's observations, I was just wondering, could social environments, not just physical environments that, as you mentioned, impose constraints to what similarly shape planning and cognition? Just as visual clutter involves occlusions and obstacles that affect how agents perceive and navigate the world, maybe we can think of social clutter as the density and complexity of social interactions, hierarchies, and information flow within a group. That kind of cognitive load would be very large, as individuals would have to track multiple others, their personalities, internal states, and social ranks, and while managing all of this while managing their own standing in the group. So a broader question is, could constraints in the social domain, like those in the physical world, have also driven the emergence of high-order cognition? And how might strategic planning differ when navigating the world as part of a social group in addition to the physical space? Lastly, we have mainly talked about prospective cognition, specifically planning, as a hallmark of intelligence. While this association between planning and intelligence is persuasive in the context of embodied agents interacting with dynamic environments, it raises important questions about how generalizable this is. Can intelligence be reduced to the capacity for planning? Is strategic behavior the hallmark of cognition or just one form of it? What might we be missing if we equate intelligence with planning? This might hold for animals where cognition is often shaped by sensory motor contingencies, but not embodied AI systems can exhibit forms of intelligence without engaging in the physical planning or environmental navigation. Thus, while planning might be a key expression of intelligence in certain ecological niches, especially those involving uncertainty or danger, it might be more accurate to view it as an instantiation of intelligence rather than a defining feature. Intelligence, specifically in artificial systems, can manifest in abstraction, learning, or coming up with creative solutions without necessarily invoking spatial foresight or action control. In this case, what would be the expected constraints of artificial systems to develop further cognition? To conclude, I want to highlight the importance of the study of individual animal behavior in naturalistic environments. Something that could be seen as simple as tracking mice, how mouse is sensing an environment and navigating it, can have profound implications to uncover the inner workings of our brain and give insights into certain aspects of animal cognition. And I'll leave you here with a quote from the book I just mentioned, which resonates beautifully with the cross-species parallels of cognition that Malcolm draws from his work. Thank you.